Myself, Mallikarjunan. I am taking the class for 10th standard science today. Good morning, students. Today we are going to discuss for 10th standard unit number 20, Breeding and Biotechnology, question answer discussion session. So, first the question paper we get, you read the questions very carefully. Tamil Nadu government has given 10 minutes time for reading the question paper. Before that, after reading the question paper, then only you want to write that examinations. Okay, now we are going to question answer discussion session, breeding and biotechnology topic. First one, choose the correct answers. Here totally there are 10 questions are there. Here question number 1, which method of crop improvement can be practiced by the farmer if he is inexperienced? A. Clonal selection, B. Mass selection, C. Pure line selection, D. Hybridisation. Answer B. Mass selection. Question number 2. Pusa komal is a disease resistant variety of dash. So B. Rice, C. Cowpea, D. Maize. So in this question number 2 answer is C. Cowpea. Next, question number 3. Himgiri developed by hybridization and selection for disease resistance against rest pathogens is a variety of. Answers: number A. Chili, number B. Maize, number C. Sugarcane, number D. Wheat. You select that answer. Answer is D. Wheat. Then question number 4. The miracle rice which saved millions of lives and celebrated its 50th birthday is. Answers are given: A. IR8, B. IR24, C. Automata 2, D. Ponni. Here correct answer is IR8. Then question number 5. Which of the following is used to produce products useful to humans by biotechnology technique? Here the answers are given: A. Enzyme from organism, B. Live organism, C. Vitamins, D. Both A and B. You select that answer is A. Enzyme from organism. Then question number 6. We can cut the DNA with the help of dash. A. Scissors, B. Restriction endonucleases, C. Knife, D. RNA-seq. Here you select that answer is B. Restriction endonucleases. Now question number 7. rDNA is a, it is the question. Answers: A. Vector DNA, B. Circular DNA, C. Recombinant of vector DNA and desired DNA, D. Satellite DNA. Here we choose the answer is C. Recombinant of vector DNA and desired DNA. Then question number 8. DNA fingerprinting is based on the principle of identifying sequences of DNA. Answers are given: A. Single stranded, B. Mutated, C. Polymorphic, D. Repetitive. You choose the answer is D. Repetitive. Then question number 9. Organisms which modify endogenous gene or a foreign gene are also known as. The answers are given: A. Transgenic organisms, B. Genetically modified, C. Mutated, D. Both A and B. You choose the answer is A. Transgenic organisms. Then question number 10. In a hexaploid with 2n equal to 6x equal to 42, the haploid n and the basic x number of chromosomes respectively are dash. The answers are given: A. N equal to 7 and x is equal to 21, B. N equal to 21 and x is equal to 21, C. N equal to 7 and x is equal to 7, D. N equal to 21 and x is equal to 7. You choose the answer N equal to 21 and x is equal to 7. Now choose the correct answers are completed. Now we are going to fill in the blanks. Question number 1. Economically important crop plants with superior quality are raised by. We think that answer in mind, write carefully. Plant breeding. Question number 2. Protein rich wheat varieties. Dash, you think the answer and write that Atlas 66. Number 3. Dash is the chemical used for doubling the chromosomes. The answer is Colchicine. Number 4. The scientific process which produces crop plants enriched with the desirable nutrients are called dash. Biofortification. Question number 5. Rice normally grows well in alluvial soil, but dash is rice variety produced by mutation breeding that grows well in saline soil. Answer automata to rice. Then question number 6. Dash technique made it possible to genetically engineer living organism. Recombinant DNA is the answer, you write it. Number 7. Restriction endonucleases cut the DNA molecule at specific positions known as dash. Answer you think of and write it, restriction site. Question number 8. Similar DNA fingerprinting is obtained for dash. We think the answer correctly and write it, identical twins. Question number 9. Dash cells are undifferentiated mass of cells. Stem cells. Actually, we want to write stem only. Stem cells are undifferentiated mass of cells. Question number 10. In gene cloning, the DNA of interest is integrated in a dash vector. So, fill in the blanks as coming in examination, either 2 mark questions or 1 mark question. Mostly it will be asked in 2 mark questions only. Okay, read carefully. Next, true or false? Rhabdobrachysa is a tetraploid man-made genus produced by Colchicine treatment. The answer is 2. Here, true or false, if want to true, no need to write that answer. Question everything they want to write, no need. You write simply true is enough. If whether it's a false statement only, if you want to write correct statement, what is the false statement given, what is the correct statement you want to write, it will becomes under the 2 mark question. So now question number 2, the process of producing an organism which more than 2 sets of chromosome is called mutation. Is a false. So you want to write correct answer. Question number 3, a group of plants produced from a single plant through vegetative or asexual reproduction are called a pure line. Here the statement is wrong. To find, you want to write the correct statement. So everything is same. At last a word is there, pure line. In the place of pure line, you want to write the clones. So statement is false. A group of plants produced from a single plant through vegetative or asexual reproduction is called clones. Number 4, iron fortified rice variety determines the protein quality of the cultivated plant. Here, this statement also false statement. You think that here the second word fortified is there. In the place of the fortified is cut the word and completely you want to write. On the place of fortified you want to write rich. Statement is false. Iron rich rice variety determines the protein quality of the cultivated plant. Question number 5. Golden rice is a hybrid. Here it's the statement is false. So the last word hybrid is there and the word will be cut and you write the correct statement. Golden rice is a genetically modified plant. Question number 6. Bt gene from bacteria can kill insects. The statement is true. You don't need for write the statement. Then question number 7. In vitro fertilization means the fertilization done inside the body. The statement is wrong. So you want to read the statement. Which word is wrong? you look that in the place of the inside, it simply you write outside. So statement is false. In vitro fertilization means the fertilization done outside the body. Number 8. DNA fingerprinting technique was developed by Alex Jeffrey. The statement is true. No need to write the statement. Again, question number 9. Molecular scissors refers to DNA ligases. The statement is false. You want to write in the place of the ligases restriction enzymes. Statement is false. Molecular scissors refers to restriction enzymes. So, true or false, it will become under 2-mark question, and fill up also come under the 2-mark question. Match the following. Next one, it is the part number 4. It is also comes under the 2-mark question. Here questions. Question number 1, Sonalika. I'll give the direct answer, semi-draft wheat. Question number 2, IR8, semi-draft rice. Question number 3, Saccharum. Answer is sugarcane. Question number 4, mung number 1. Answer, Phaseolus mungo. Question number 5, TMV2. Answer, groundnut. Question number 6, insulin. Answer, first hormone produced using rDNA technique. Question number 7, Bt toxin. Answer, Bacillus thuringiensis. Question number 8, golden rice. Answer, beta carotene. Next one, assertion and reasoning. Here also the same like choose the correct answer. You want to correct the assertion is given, reason also given. Whether there are 4 choose the correct answers as given. Number 1, assertion is correct and reason is wrong. Number 2, reason is correct and the assertion is wrong. Number 3, both assertion and reason is correct. Number 4, number D, both assertion and reason is wrong. This is the question. Assertion and reasoning. Understand the assertion statement, justify the reason given, and choose the correct choice. Question number 1. Assertion. Hybrid is a superior than either of its parents. Reason. Hybrid vigor is lost upon inbreeding. Answer, C. Both assertion and reason is correct. Now, question number 2. Assertion. Colchicine reduces the chromosome number. Reason. It promotes the movement of sister chromatids to the opposite poles. You write that answer. Answer is, choose the correct one. D is the answer. Both assertion and reason is wrong. Question number 3. Assertion. rDNA is superior over hybridisation techniques. Reason. Desired genes are inserted without introducing the undesirable genes in target organisms. Which one is the answer? You choose that. Answer is A. Assertion is correct and reason is wrong. So, you complete the assertion and reason. Now, you read answer in a short sentence. Okay. Question number 1. Give the name of wheat variety having higher dietary fiber and protein. Triticale 6N. Answer is Triticale 6N. Question number 2. Semi-draft varieties are introduced in rice. This was made possible by the presence of drafting gene in rice. Name this drafting gene. Answer, defective gibberellins 20 oxidase gene. Now, question number 3. Define genetic engineering. Answer, genetic engineering is the manipulation and transfer of genes from one organism to another organisms to create in a new DNA called as recombinant DNA. Simply you want to write in brackets rDNA. Question number 4, it's a very famous question in exams. Name the types of stem cells. Answer, there are 2 types of these stem cells. Answer number 1, embryonic stem cells. Number 2, adult or somatic stem cells. Question number 5. What are transgenic organisms? So, answer you want to write: plants or animals expressing a modified endogenous gene or a foreign gene are also known as transgenic organisms. Question number 6. State the importance of biofertilizer. Here I will give the answer for points like structure. There are 2 points and one example is there. First point: biofertilizers are easy to produce in abundance and are available at low cost to the marginal farmer. Point number 2. It is increased the soil fertility without causing any damage to the soil. Examples: Rhizobium, Actobacter. So, answer in a sentence completed. Now we are going for short answer questions. Question number 1. Discuss the method of breeding for disease resistance. You want to write the answer. Plant diseases are caused by pathogens like viruses, bacteria and fungi. This affects crop yield. Hence, it is important to develop disease resistant varieties of crops that would increase the yield and reduce the use of fungicides and bactericides. Example: Himgiri wheat is resistant to leaf and stripe rust, hill bunt diseases. Question number 2. Name 3 improved characteristics of wheat that help India to achieve high productivity. Here also given the points. There are 3 points given. Point number 1, Atlas 66. It is used protein rich wheat variety. Point number 2, Himgiri, resistant to diseases like leaf and stripe, rust, hill bunt. Point number 3, Sonalika, high yielding semi-dwarf wheat variety. Now question number 3. Name 2 maize hybrids rich in amino acids lysine. Answer just 2 points only. Point number 1, Protina. Point number 2, Sakti. Now question number 4. Distinguishing between. There are 2 questions as given. Question number A, somatic gene therapy and germ line gene therapy. The answer for this: somatic gene therapy. It is the replacement of defective gene in somatic cells, bracket body cells. Germ line gene therapy. It is the replacement of defective gene in germ cell. Only simple thing. First one you want to write somatic cells. Second place you want to write germ cell. In germ you want to write the example egg and sperm. Question number B. Undifferentiated cells. Stem cells are unspecialized mass of cells that having variable potency is called undifferentiated cells. Example: umbilical cord. Differentiated cells. Our body is composed of some specialized cell types that can out specific functions are as differentiated cells. Example: pancreatic cell to secrete to insulin. Next, question number 5. State the applications of DNA fingerprints technique. DNA fingerprinting technique is widely used in forensic applications like crime investigation, such as identifying the culprit. It is used for paternity testing in case of disputes. It helps in the study of genetic diversity of population, evolution and speciation. Question number 6. How are stem cells useful in regenerative process? Answer: sometimes cells, tissues and organs in the body may be permanent damage or lost due to genetic condition or disease or injury. In such situations, stem cells are used for the treatment of diseases, which is called stem cell therapy. In treating neurodegenerative disorders like Parkinson's disease and Alzheimer's disease, neuronal stem cells can be used to replace the damaged or lost neurons. Question number 7. Differentiate between outbreeding and inbreeding. Here, first one, outbreeding. Crops between two different species, which is desirable features of economic value, are mated. The hybrids are stronger and vigorous than their parents. Example, mule. Inbreeding. Mating of closely related animals within the same breed for 4 to 6 generations. It helps in the accumulation of superior genes and elimination of genes which are undesirable. Example, hersalde. So, next one, long answer questions. So, in this lesson, long answer questions are given directly, 7-mark questions. So, direct 7-mark question, only one question will be asked. Here, very important questions are there. First one, what are the effects of hybrid vigor in animals? Hybrid vigor. The superiority of the hybrid obtained by crossbreeding is called heterozygous or hybrid vigor. Efforts of hybrid vigor in animals. You want to write 4 points. Point number 1, increased production of milk by cattle. Point number 2, increased production of eggs by poultry. Point number 3, high quality of meat is produced. Point number 4, increased growth rate in domesticated animals. It is the answer for question number 1. Then question number 2, describe mutation breeding with an example. Mutation. Here also given 5 points. Point number 1, mutation is defined as the sudden heritable change in the nucleotide sequence of DNA is an organism. Point number 2, it is process by which genetic variations are created, which in turn brings about changes in the organism. Point number 3, the organism which undergoes mutations is called mutant. Point number 4, the factors which induce mutations are known as mutagens or mutagenic agents. Point number 5, mutagens are of 2 types, namely physical mutagens and chemical mutagens. Now we want to write physical mutagens. Radiations like X-rays, alpha rays, beta rays, gamma rays, UV rays, temperature, etc., which induce mutations are called physical mutagens. Then chemical mutagens. Chemical substances that induce mutations are called chemical mutagens. Example, mustard gas and nitrous acid. The utilization of induced mutations in crop improvement is called mutation breeding. Achievements of mutation breeding. Number 1, Sarbati sonara wheat produced from Sonara 64 by using gamma rays. Point number 2, Automata 2 rice with saline tolerance and pest resistance. Point number 3, groundnuts with thick shells. So it is the answer. Now we go to question number 3. Biofortification may help in removing hidden hunger. How? Here also given point like structure. Number 1, biofortification is the scientific process of developing croplands enriched with high levels of desirable nutrients like vitamins, protein and minerals. Examples. Number 2 point is examples. Here there are 4 examples as given. A point, Protina, Sakti and Ratna are lysine rich make hybrids. In brackets you want to write developed in India. Point number B, Atlas 66, a protein rich wheat variety. Point number C, iron rich fortified rice variety. Point number D, vitamin A enriched carrots, pumpkin and spinach. So in this way, biofortification may help in removing hidden hunger. Now question number 4. Here, with a neat label diagram, explain the techniques involved in gene cloning. Here label diagram, diagram is there, I will explain the diagram later. First one, you want to write the statements clearly. First one, what is gene cloning? First you want to write gene cloning is what? There are 2 points is given. Point number 1, the carbon copy of an individual is often called a clone. However, more appropriately, a clone means to make a genetically exact copy of an organism. Point number 2, in gene cloning, a gene or a piece of DNA fragment is inserted into a bacterial cell where DNA will be multiplied, or otherwise you want to write copied in brackets as the cell divides. Then you want to write techniques involved in gene cloning. There are 5 points. You want to write point wise. Point number 1, isolation of desired DNA fragment by using restriction enzymes. Point number 2, insertion of the DNA fragment into a suitable vector to make rDNA. Point number 3, transfer of rDNA into a bacterial host cell. Point number 4, selection and multiplication of recombinant host cell to get a clone. Point number 5, expression of the cloned gene in host cell. So these are the points you want to write. Here simple label diagram. So label diagram is nothing. So very easiest diagram, you draw like a egg, that means of elliptical shape egg, you know very well. On that egg you write a ribbon like structure. Inside one ribbon like structure is there. Ribbon like structure is DNA. What is the structure of DNA? You know, double stranded helix. So just a coil like structure, you draw inside of the egg just small coil, that is an human egg, inside a small DNA. Now after that DNA will be come out. So you come out the DNA will be gene, DNA will be come out, that the DNA will be right. Okay, so you draw the diagram in book, you select the correctly and you draw them. So students, please read carefully the answers of long questions, because this one of the long question will become under 7 mark direct question in public examinations. Please don't forget it. It is the easiest answers for public examination you want to write directly. Otherwise, 7 mark questions you give A, B, C like parts like structure. No need for the parts like structure. It is the direct question. You read carefully in this long answer questions, you get 1 7 mark question. Definitely you are getting 1 7 mark question. Otherwise you are getting for and 2 mark question and 4 mark question 1. Okay, thank you very much.